Bill Andrews from SENS Sciences. His talk is entitled Small Molecule Stimulators of Telomerase.
Well, first, thank you, Aubrey DeGray, for organizing this conference. I really love living, and I think that I want to live as long and healthy as possible. And meetings like this is going to help that become a reality. So thank you. And also thank you for inviting me. So my title is Stimulators of Telomerase. It says in the package that it's small molecule stimulators of telomerase, but I'll be talking about more than just small stimulators. So there's a lot of diseases. There's been like thousands of publications now showing that telomeres might be associated, at least the length of telomeres might be associated with almost every disease you can imagine. Anything that involves cell division seems to play some role in causing diseases associated with telomere shorting. And so the general conclusion from all these studies leads to this one thing: bad things happen when telomeres get short. And so what we are working on is, can we link them? And so for those who actually have not a lot of experience in telomere biology, I first want to address the question of what is a telomere? And so to do this, we need to zoom in on a human being and into its 100 trillion cells, and zoom in even further into the nucleus of one of these cells, and zoom in further to the chromosomes. And the telomeres are actually the very tips of these chromosomes. The telomere is essentially about 15,000 bases in length, at least when we're first conceived. And then as our body starts to develop, the telomeres shorten. And then when cells divide, telomeres get shorter. And when they get down to about 10,000 bases, that's when we're born. As our bodies develop even further, and we grow up and our cells divide and divide, and telomeres get shorter, when they get down to about 5,000 bases, that's when we typically die of old age. So let me review this. If we are conceived at 15,000 bases, we're born at about 10,000 bases, and we typically die of old age at about 5,000 bases. And so telomere length controls aging. That's the theory. Okay? Anybody that thinks that we know that is misconceived. Okay, so right now it's just a theory. There's no data whatsoever that really says that telomere length controls aging. But it's a model that we're working on, and we want to see if there's something that can be done. There's a catch-22 that's always been involved, and that's even when I worked at Geron Corporation 20 years ago. The president of Geron used to always ask me, why are we trying to find ways to lengthen telomeres when we don't even know if telomeres have anything to do with aging yet? And I said, well, the only way we're going to find out is to find a way to lengthen telomeres, and that's the catch-22. So that's what we've been working on. So at SENS Sciences, our goal is to answer this question: does telomere cure aging or die trying? Answer the question: does telomere length actually affect aging? And then if it does, we're going to do something about it. So misconception number 2 is that only telomeres control aging. There's a lot of theories about why we age. And because of the fact that I love living, as I said before, I practice every theory I can to make certain that I extend my lifespan and health span as long as possible. Some of these you might see are the mitochondrial dysfunction theory, the oxidative stress theory, and the telomere theory of aging, which seem to be the most common ones talked about nowadays. But I like to think of these as multiple sticks of dynamite that are burning inside of all of our cells. And so each aging theory is a stick of dynamite. But what really matters is which stick of dynamite has the shortest fuse. And we don't really know. But at SENS Sciences, we're kind of hedging on the idea that it's our telomeres. The telomere shortening is the shortest fuse in humans, at least. That doesn't mean that there's not other kinds of aging occurring inside of our body. If we put out that fuse for telomeres, we're still going to have something else aging us. And is it oxidative stress, mitochondrial dysfunction? We don't really know yet. So misconception number 3, and I'll be talking about a few misconceptions here, is telomere shortening couldn't possibly have anything to do with aging because mice don't have telomere shortening, but they still age. And to address that, I'd like to go back to the dynamite model of telomere, or of aging. And in humans, we have, I'm going to just show two sticks of dynamite now. One is aging one, let's say, is telomeres, and aging two is something else. We don't know what that is, oxidative stress, mitochondrial dysfunction, whatever. Okay, so in mice, this might be exactly the opposite. In fact, we kind of believe it is the opposite because we know that mice don't have telomere shortening except when they start to get older. And so the fuse for telomere shortening is much longer. They still have this other stick of dynamite that is actually causing their aging. Again, mitochondrial dysfunction, oxidative stress. Those are the best guesses. All right, now, I think most of us have seen the paper recently from Rhonda Pinnell's lab at Harvard, where he was actually able to make mice younger, or at least appear more youthful and healthier. He took old mice and made them more youthful and healthier by lengthening their telomeres. This got a lot of press. But what I'd like to do is I'd like to explain this particular experiment in terms of the sticks of dynamite again. Notice in the case of mice, the mice normally have this very long fuse for telomere short. What Rhonda Pinnell did is he first engineered these mice so that fuse was short. And now all of a sudden these mice started aging by telomere short. And as a result, they essentially died at about 1 year of age. He then, so when these mice got to be very old, he then lengthened the telomeres and then found that these mice actually started acting younger, started looking younger, a lot of different odors were behaving better, and he was able to show that this was due to the lengthening of the telomeres. Whether this was aging wasn't really shown, but he was able to show that the mice became more youthful and healthier. Now, as it turns out, several labs have now looked at animal species all over the world, looking to see how common what's called telomere replicative senescence is. And that's where senescence occurring by telomere shortening. And it turns out to be really rare. Okay, almost all the animal species in the world, fish, birds, reptiles, they all age by other mechanisms besides telomere shortening. And then it turns out it's only found in mammals, but not all mammals. Rodents, for instance, do not age by, like as I said, mice don't age by telomere shortening. And in fact, the only mammals that have been shown to actually age by telomere shortening, or at least have replicative senescence, telomere replicative senescence, are dogs, cats, horses, primates, deer, sheep, and pigs. And it's good to see our favorite pets are up there too. All right, so this takes me to the next part of my talk. Can we lengthen them? And telomerase, this is a cartoon of telomerase. The green is showing the DNA as a double helix. Telomerase is bound to the very end of this telomere and lengthening it. And that's what telomerase does. It lengthens telomeres. Unfortunately, it's only found in our reproductive cells. The gene is turned off in all of our other cells. And, that's a whole other lecture as to why that possibly is. But at Sierra Sciences, we are doing high-throughput drug screening, or at least, since we're not just doing drugs anymore, we're doing high-throughput screening, trying to find synthetic chemicals or natural substances that will get inside the cells and turn this telomerase gene on as a way of lengthening telomeres. So we're using high-throughput screening robots, shown here. We have 2 of them there. And this is a typical example of one of our screens. If you look, the y-axis is showing the amount of H-TERT expression. H-TERT is the name of the protein component of telomerase, which is the rate limiting step. So in order to turn telomerase on in the cell, you need to turn the H-TERT gene on. Over on the far left, you can see that we have negative controls, positive controls, and we have 2 sets of negative controls and 1 set of positive controls. And then occasionally, these are different synthetic chemicals in this case that we're screening. Occasionally we find one that actually gives production of the H-TERT mRNA in the cell. And you can see we do them all in quadruplicate, just to verify that we don't have some positive. And and we call those a hit. When we get a hit like that, we see hits about one in every 2,000 different things that we screen. When we get a hit, we then run the PCR product onto a electrophoresis gel, and we want to verify that we see a band of the right size, which says that we're not amplifying something else that looks like telomerase. We then do telomerase activity assays, which normally are called trap assays. And this is an electrophoresis gel showing just how we analyze one of our hits. This one is C00576884. And on the far left, we run HeLa cells. HeLa cells are a cancer cell line. And they produce a low-level amount of telomerase, but enough telomerase to actually make themselves immortal, to keep the telomeres from shortening any further than they already are. So we have X amount of cells in the very first lane there. In the next lane, we have 5% as much of those cells. So we have two different quantitation markers, essentially. The next lane shows our normal cell line, which is, in this case, we're using MRC5 fibroblasts, human MRC5 fibroblasts. And you can see that there's no ladder. So telomerase in the trap assay is identified by seeing a ladder of bands. And in that case, in that untreated lane, you see no bands. But in all 6 replicates of testing C00576884, we see a ladder, and we see that it's about equivalent to what 5% of HeLa does. So that means that we are producing enough telomerase from this compound to produce enough telomerase to be about 5% of what HeLa is, and hopefully that's about 5% of what it will take to make a human cell immortal, or at least not have replicated senescence. Well, one major thing that we discovered through all this, and this has been predicted before, but I don't think anybody's ever able to show this, is that the rate-limiting step of telomerase expression is totally at the mRNA. So once we figure out a way to turn on the messenger RNA, turn on the promoter to produce that messenger RNA, all the different steps of expression, transcription elongation, translation, post-translational modifications, transport of the protein, all that seems to work, and we get activity. So it seems like the main thing you got to do to turn on telomerase is to turn on the telomerase promoter. And so far, we have screened 257,163 chemicals and natural substances. And from that, we have found 57 families of hits. 39 of those are synthetic chemicals, where when we find different hits, we find sometimes they look very similar. One might have a fluorine versus a bromine on the chemical. That way we just put them into the same family. We have 39 families of chemicals, and the rest of them are natural substances. When we get a hit, we try to give it a score by looking at, like I was showing before, that C00576884 was 5% of HeLa. That would mean it has a score of 5. And the scale shows 0 to 100, where 100 means that it's got the same amount of telomerase activity as a HeLa cell. Well, 0 would be essentially the equivalent to a clock ticking at normal speeds. This is ticking a little faster than normal, but in order to show my next slides, I'm going to have to have it just ticking faster. But this is now, without any telomerase, let's say, how much a cell would essentially age, or its replicated senescence would occur. We have found hits now, both chemical and synthetic, that produce enough telomerase to essentially slow this clock down. We have synthetic or natural substances that score a 3 on the scale, and we have synthetic chemicals from our screening, random screening, that score a 6. With the synthetic chemicals, at least, we've been able to do medicinal chemistry to design even better chemicals from the information we learned from our random screening. And we've been able to get chemicals up to 16 now. So we have things that are 16% of the way to actually being where we predict replicated senescence will stop, at least telomere replicated senescence. Now, in the next year to 3 years, we actually feel that we can actually get to a point of having 100, where we can actually produce enough telomerase to stop the clock. And then after that, we hope to knock the roof off that, and actually get things that will actually reverse it. And the only reason that we believe this is possible is because of Rhonda Dennell's recent experiments in Harvard. We've always thought it might be possible, but at least the best proof of concept that this could actually be a reality is from Rhonda Dennell's experiments. So So at this point, I'd like to talk about clinical studies. And first, I gotta say, there aren't very many clinical studies because there aren't very many things available now that we can take that can lengthen pectin turn on telomeres. But the very first thing that's come on the market is called TA65, marketed by TA Sciences. And we did a clinical study on that a few years ago. Unfortunately, it wasn't a double-blind, placebo-controlled experiment, and it really wasn't from a random population. We were only provided with blood samples from people that had already been taking TA65. But we did get some interesting results from that. Well, besides the fact that we did see an improvement in immune functions, the more important thing, or at least the significant thing that I want to present here, is that we did see that telomeres were getting lengthened. At least we could see that the shortest telomeres were getting lengthened. What this is showing on the y-axis now is the number of short telomeres that are found in a particular blood sample from a person. And these are 12 different subjects that were tested. These subjects had all been on TA65 for more than a year. And so the first bar, the black bar in each case, is before they went on the drug, or drug, I better not call it a drug, the natural product TA65. And the gray bar is at least 1 to 4 years afterwards. And what we see is in 10 out of the 12 cases, we actually saw a reduction in the number of short telomeres. This is published in Rejuvenation Research 2010. And the only 2 cases where we didn't see a reduction in the number of short telomeres was the 2 cases here that had the lowest amount of short telomeres to begin with. Those were the 2 youngest people. And it might be that they just didn't have enough short telomeres to actually really get an accurate measurement there. But in all the other cases, the number of short telomeres got reduced in the patients taking TA65. Again, this was not a double-blind placebo-controlled, so there was no placebo group here. And this isn't a random population because it came from people that actually were concerned about their health enough that they actually went and purchased TA65. Now, another product has just been launched in the last 2 weeks. It's marketed by Isagenix. And we have a clinical trial that's just now getting underway. In this case, it will be a double-blind placebo-controlled experiment, and it will come up from a random population. There will be 30 random subjects included in this experiment. They'll all be pre-screened for short telomeres, since the main thing we'll be measuring is the production of short telomeres. We don't want to be using subjects that don't have any short telomeres to measure, such as very young people. So it'll probably be mostly people will be over 65. We'll be measuring baseline, 3 months, 6 months, and 12 months. And adverse events will be tracked, so any kind of health-related issues that might surface as a result of telomerase induction will be measured. These are a list of the different things that we'll be studying. I'm not going to go through them one after another. Feel free to look at them, and if anybody is interested in talking to me about it, it's not too late to add another test. And so this is just some of them, and the other ones are listed right here. This study is supposed to get underway in September. We'll also be photographing people, faces, hands, because one of the things that was noticed by a lot of people taking TA65 is age spots were disappearing, but nobody had any documentation of that. So we'll try to get in on that stuff too. All right, so since I am in the module today called cancer, I thought I should talk about cancer. It's well known that 85 to 95% of all cancers express telomerase. Well, this means, therefore, that telomerase must cause cancer. And I want to present this as a misconception. It's not necessary. It could also be just as easily explained as cancer caused telomerase suppression. So telomerase doesn't necessarily cause cancer. Cancer could be causing telomerase. And we don't know. And we don't know because correlation does not mean causation. What I want to do is present some facts and stuff about this, suggest that we really don't have data saying that telomerase causes cancer. It just is something that actually became a common thing to say, and I rank it as essentially a rumor. There's been no scientific publications that show that telomerase causes cancer. There has been a few publications suggesting that telomerase causes cancer. And all of these have been in mice. And there hasn't been any publication since 2005. And even the authors that published those papers are now publishing papers saying something else, which I'll come to. So the problems with the papers that have been saying that telomerase causes cancer is there's such a thing as artifacts of overexpression. Anybody who has a lot of experience, like I had years ago, when my job at a company called Burlex Biosciences was to overexpress proteins for production reasons, you know that overexpression of just about any gene is going to wreak havoc on expression throughout the genome. So expression of any gene can cause problems. Another problem is insertional mutagenesis. And all experiments that have been looked at so far, they've inserted the genes into the chromosomes. And we know that expression can be affected by point mutations that are like hundreds of thousands of bases away from the gene that you're looking at. And so it's really hard to insert a gene into a chromosome without having some kind of effect on gene expression somewhere else on the chromosome and even on other chromosomes. Now, mice are also very cancer prone, which is something that kind of like makes this data a little less interpretable. But you notice that in some of the papers that people have read these, people have made cancer-resistant mice in order to study the effects of telomerase. But since mice are very cancer-prone, these telomerase resistance effects that they did probably made mice about as cancer-resistant as humans already are. And there's also been papers on other activities of telomerase, and that's okay, as far as I'm concerned. It's okay if telomerase is doing other things besides lengthening telomeres, unless they're also causing some problems. And hopefully, if they are causing problems, we can block those activities without blocking the activities that we really are interested in, and that's the telomere lengthening. Now, there has been, years ago, many studies that have been done, just about everything imaginable, to test whether or not telomerase does cause cancer. Miscellaneous soft auger assays, contact inhibition assays have been done back in approximately year 2000, and absolutely nothing was detected that suggested telomerase could cause cancer in any way possible. But now, as I was saying, the authors that we're publishing that telomerase might be causing cancer, the tune has changed a lot since 2005. Now there's a lot of data saying that short telomeres cause cancer. For every paper that ever suggested that telomerase causes cancer, there's now 20 papers that are saying that short telomeres cause cancer. And what's happening is when telomeres get short, chromosome rearrangements occur and mutations occur that can lead to cancer. So telomeres, remember, are something that protect the tips of the chromosomes, and when they get short, the chromosomes can rearrange and fall apart. And all these kind of mutations can cause all the different genes that can be mutated that are required to cause cancer. There are some people that say that cancer requires up to 10 to 12 mutations. And it might be that all these mutations pretty much occur all of a sudden when telomeres get short. The immune system becomes weaker and less capable of fighting cancer when telomeres get shorter. We know this from the AIDS. When people were first getting the AIDS virus back in the early 80s, doctors were really surprised that these patients had no T cells. But that was because their T cells had disappeared due to telomere shortening, and their immune systems became very weak, and they started getting a lot of different diseases like sarcoma, typical cancer. So the message here is what we really want to do is keep telomeres long, and telomerase keeps telomeres long. And so telomerase might be a much better player than a lot of the rumors going around that it's something that can cause cancer. It might be the best way to actually fight and cure cancer. So cancer summary, short telomeres cause cancer and disease and decrease our ability to fight cancer, and telomeres can keep telomeres long. I'd like to end with one little kind of interesting observation. And that's that the National Institutes of Health reports that if you take 1,000 people at random, age 60 to 64, and look at them for a year, you'll find that 13 of them get cancer. That's pretty much the statistics for that age group right now. But in the case of TA65, that looked at a lot more than 1,000 people at that age group, they've actually had no reported cases of cancer yet. What this means, I don't know because it's not a real clinical study, but it is an interesting observation. It is mentioned in this publication. in Rejuvenation Research 2010. And I want to thank you very much. And any questions?
I think we have time for one question.
Is there sufficient epidemiological evidence in human beings that, when adjusted for age, individuals with short telomeres live shorter?
There are some studies from, okay, some not really in detail, as detailed as you want, but there is data saying that people with shorter telomeres do live shorter lives. Okay, again, does that mean that telomeres have anything to do with aging? I'm not really saying that. And my goal is to find something that's going to extend my lifespan. I'm not just going to believe in telomere biology just because it's a good story. So I want to find a real thing that works. And I don't think that that data is actually showing that telomere shortening affects aging. Could be other explanations. Can I do it over time?
I'm having a hard time figuring out how to formulate this question, but yes, we know that telomeres shorten. We do know that that's an information degradation. What is the mechanism by which they are shortened? In other words, like a car, they hit the wall, is there a samurai that cuts them off? What's doing it?
Well, first of all, degradation is really the wrong word. Degradation does play some role, at least by free radicals, in causing telomere shortening. The telomere shortening is mostly caused by the lack of the ability of the cell to replicate the very tips of the chromosome. So every time the DNA replicates, the telomeres can't fill in all the way to the very end. So the telomere is short. So it's not really degradation that shortens telomeres. Every time a cell divides, the telomeres get about 50 to 100 bases shorter.
You're saying what happens, I'm asking
So,
why.
okay, so the question is, why does telomere shorten? It's possibly the chromosomal rearrangements. There's several models. Okay, one is chromatin-related models, but the model that I like the best, I don't know if it really explains it, but if you know a lot about how genes express, you can have a cancer sequence that's like 10,000 bases away from the promoter, and it can actually fold over and interact with the promoter to turn it on. Think of telomeres as enhancer sequences. And so they actually fold over onto the rest of the chromosome to interact with other genes, turning them on and off, just like enhancers. Now, if the telomeres get shorter, they're less capable of reaching all those genes. Another explanation is simply the thing I was talking about, the chromosomal rearrangements and mutations. It might be that the average length of the telomere has nothing to do with anything. It might be just the total number of short telomeres that we have. And so the average is going to correlate to the number of short telomeres. So when we get short telomeres in a cell, then we have chromosomal rearrangements and mutations, and that can lead to damage to cells that come across the region. You see either one of those answer your question. Because nobody knows the answers. Okay, that's what we're trying to tell you is that there's models to explain it.
Okay, to keep on the time frame that we have this evening, we're going to move on to the next speaker. We'd like to thank Dr. Andrews.